Welcome to another exciting episode of the BCI podcast. Today, we have the privilege of hosting a distinguished guest, Professor Anil Koul, a renowned leader in the field of global public health and infectious disease discovery. Professor Koul currently serves as the Vice President and Head of the Global Public Health and Infectious Disease Discovery Unit at Johnson & Johnson Pharmaceuticals, operating from Belgium. With over 20 years of experience in drug discovery, development, and scientific innovation, Professor Koul has made remarkable contributions to the field. Previously, he held the position of the director at the Institute of Microbial Technology, a premier biotechnology lab under the Ministry of Science and Technology, Government of India. His exceptional leadership and dedication to advancing research have positioned him as a key influencer in the industry. Professor Koul's expertise extends beyond his corporate role. He has recently been appointed as an adjunct professor of translational research at the esteemed London School of Hygiene and Tropical Medicine. This appointment reflects his commitment to driving translational TB discovery research, furthering our understanding and treatment of tuberculosis. One of Professor Koul's most significant achievements is his pivotal role in the discovery and development of Bedaquiline, a groundbreaking drug for the drug-resistant tuberculosis. Bedaquiline has been conditionally approved in over 64 countries and has made a remarkable impact on the lives of more than 300,000 TB patients. Recognized for his contributions, Professor Koul has received prestigious awards such as the Sun Pharma Research Award and the Swiss Tuberculosis Prize. Beyond this groundbreaking research, Professor Koul has published in leading scientific journals and holds more than 25 international patents. His expertise in moving drugs from discovery into clinical development is evident through his involvement in the progress of 3 other drug candidates, including one currently undergoing phase 3 clinical testing for respiratory viral infections. Professor Koul's passion for global public health and his drive to bridge the gap between scientific innovation and neglected diseases have shaped his distinguished career. His work spans across 3 continents, and he currently serves on the board of directors of Janssen Pharmaceuticals, Belgium, and the scientific advisory board of the Council of Scientific and Industrial Research, India. We are truly honored to have Professor Anil Koul on our podcast today. Welcome to the BCI podcast, Professor Koul, and thank you so much for joining us today.
Thank you, Dr. Manish. It's so nice to be with you. It's wonderful to see you also.
Amazing. So let's dive in straight away. I'll start with, what inspired you to pursue a career in science and how did you get started in the field?
Oh, that's a fantastic question. I think my interest in science started when I was a kid in the school. As you might remember, 1980s, early and mid 1980s was a phenomenal phase of biology. It was a phase when people started cloning genes. It was a phase when we had a new discovery in terms of recombinant DNA technology. At that time in the school, I was still in the secondary school. At that time, we had a concept came across in the classroom, which was around jumping genes. So we, with all naivety, me especially, and some of my friends, with all naivety, thought that these genes are jumping from one place to another. And it fascinated me as I thought of what exactly are these genes, why do they need to jump? And eventually became what we call genetic drifts and all the genome rearrangements that happen. But as a school kid, the concept of that you can cut and paste the genes, the gene cloning, which was in the infancy at that time, it really, really stimulated me to go in the field of biology, to go in the field of biotechnology, and a subsequent application in terms of in medicine as such. So I can tell you that I grew in a school with all these fascinations of science and miracles and the promise of biotechnology to shape human health. And that's how I stuck to the biology and stuck to the biotechnology of biomolecular medicine.
Amazing, amazing. So, I would be more interested in knowing your educational journey and perhaps also, what apart from science fascinated you? Like, was there any childhood dreams of yours or something like that? Please highlight your educational journey and about the dreams.
Yeah, I think when I was in the school, in the secondary school, there used to be an advertisement coming out in radio at that time. It was about and more directed towards the girl child.
Hmm.
It was from the government itself, and the advertisement was that if you don't want to be an engineer, you can be a genetic engineer.
Oh,
This was this was really fascinating for me because first and foremost, it gave me a perspective that there's something else you can do apart from the conventional story of being a doctor or an engineer, which most of the Indian families have, that the conventional route of success. But this was something very fascinating. I don't know who designed that advertisement, and must have been very, very creative guy. But every morning I used to listen it at around 9 a.m. Before the school time, and it somehow and that word genetic engineering stuck with me and brought the entire fascination in terms of the gene cloning and in terms of the genetics, in terms of genetic manipulation, in terms of all the possibilities of biotechnology. So that was the beginning of it. Eventually, and luckily, I should say, it stayed with me, that interest, that fascination. And in 1990s, a strange thing happened, or an unfortunate thing happened. We had to leave J&K, Jammu and Kashmir, where I was born. I was born in Srinagar. I did my entire schooling in Srinagar, which is part of Jammu and Kashmir. So with all the civil,
well, which school did you went, by the way? Like, in
it
J.
was called Hindu High School,
Hmm.
and but you all know the
The
issue
tragic situation that
that
happened.
reason happened in 1990s, and that evolved, and because of that, we had to leave our home. We the whole community suffered a loss of the loss of being uprooted from the place where you had generations and generations of existence. So that tragic event still shaped my life a lot and shaped all of our lives a lot. And that migration, that trauma, that pain stayed in terms of getting uprooted from the place where you have been living for generations. So after that, after the schooling, I went to Delhi University. I had an opportunity to option of studying medicine back in J&K or in Jammu, which, as I said, that I somehow wanted to rebel against the conventional wisdom or the conventional career choices of being a doctor or an engineer. So I went to Delhi University and started studying biology. And Delhi University being an amazing place, it shaped me for the rest of the life. And I did my bachelor's in Delhi University. I did my master's in Delhi University, and then that eventually helped me to transition into a PhD at Max Planck Institute of Biochemistry in Martin Street in Germany.
So tell us
So that
more about this journey. Like, how did you end up in Germany?
So it's very interesting because after my master's, I joined an institution called CET at that time, Center for Biochemical Technology, which is part of the Council for Scientific and Industrial Research.
And now it's IGI. Yeah.
Now it's called IGIB, Institute of Genomics and Integrated Biology, as once again part of the CSR group of organizations for Government of India. So at IGIB, I had a fantastic guide His name was Dr. Yoginder Singh. One of the phenomenal persons I have met in my life, not only as a scientist, but as a human being. I think the human aspect of him was really amazing. So we started doing what we call TB discovery in IGIB in his lab. So we started looking at signaling molecules in Mycobacterium tuberculosis with the aim that at one day we will be able to target them for therapeutic applications. Dr. Singh once came, I still remember the day, he came to me and said that, look, Anil, all our expertise, what the lab is doing is in anthrax.
Mm-hmm.
His expertise was his anthrax biology. And you started looking at TB, so it's relatively new in the lab. So I don't think it's a good idea to move ahead for looking at these kinases and phosphatases in Mycobacterium tuberculosis when we have no evidence. We don't know whether they even exist. We don't know what we are looking for. And I'm not sure that this is the right proposition. I said, but look, this is a chance to do something completely different, which nobody has done before. So why don't we stick to it? He said that if we want to still do it, let's see if there are other collaborative partners or labs which have more established kinase biology and phosphatase assays and platforms available. I said, okay, I'm fine with that. I was a young student. I had my entire life before me to explore and also failed. So what Dr. Singh said that, okay, let's try to look for new collaborative partners. So what I did for the next 1 month, I looked at all labs across the world who are doing kinases and vertexes in all naivety.
So globally.
And in the end, I landed up with a publication. At that time, of course, there was limited use of the Google, there was no Google at that time. So what we did, we out of PubMed at that time.
Mm-hmm. Mm-hmm.
So what we landed up was, I landed up with a publication in Nature by a group in Germany, which was Max Planck Institute of Biochemistry, with the last author, Professor Axel Ulrich, and it was all about kinases and the biology in cancer. So a role of receptor tyrosine kinases in oncology. Yeah, with all naivety and with all ignorance, I wrote to Professor Ulrich that I'm interested in discovering kinases in Mycobacterium tuberculosis. Are you also interested in it? It's a common interest. Your interest is in oncology, my interest is in TB.
So you as a student wrote him for a collaboration, right?
I, as a student, wrote for a collaboration because Professor Axel Ulrich said that I am also very interested in TB and microbacterial kinases. So why don't we collaborate? Why don't you come out? And that's how I landed up eventually with all the fellowships and all the paperwork and all the interviews that had to happen. It happened, and as destiny would have been, I landed up in one of the best, most famous labs in the world in kinase biology.
Amazing. And, tell us more about your, days in Germany, what science you pursued in the lab. And then also later on, please tell us about, how did you transition to the R&D role in J&J, about that transition. Yeah.
Yeah, I think being at Max Planck, being in the lab, which was at the forefront of kinase biology in cancer, which was at the forefront of transforming the cancer, because Axel Ulrich was the person who did a fundamental discoveries in discovering the drug which is called Herceptin, which is one of the major drugs for treatment of breast cancer. It's an antibody against a HER2 receptor, and he had a fundamental role for which he was recognized globally for that. for that discovery and several other phenomenal discoveries. But within the lab also, his passion was, how do we change the global public health? How do we change the field of TB? He had seen the innovation happening in cancer, but for disease like TB, we all knew that there was no drug available for last 45 years. So I started in a lab. Everybody else was looking at cancer biology, and I was the only one who was looking at TB using. But I had the resources of all the tools and techniques available to me in a cancer biology environment.
Mm-hmm.
But I think it gave me fundamental insights into how we should address a problem looking at multidimensional. And at the same time, it also, since Excel was coming from industry background, he had a gene and tech role, and he went to the academia. So it also, he was very keen on looking at the translational aspects of basic biology drug discoveries. And he was a serial entrepreneur. He founded several companies. And yeah, that put me in the forefront of that. No, we need to do something for TB. We need to discover new drugs, and there must be other ways of a drug discovery for TB. And that's why I went from looking from kinases or host targets for TB into more into what I call some very novel targets when I joined J&J in 2003 or 2004.
And how did this transition happen? Like, you're somebody really interested in science. So why didn't you pursue the classical PhD, postdoc, professorship route rather than this industrial route? Like, how did this transition happen?
So at that time, Excel said that I always thought that oncology is much more privileged and career choice than TB.
Mm-hmm.
He said, no, let's focus on the kinases of TB. Let's see how we can transform TB. You came from India to this lab to do TB research, so you should not get swayed away by
the glamour of oncology.
By the glamour of oncology, by the tools available for oncology. You should stay in TB. Mm-hmm. And I think his persuasion was very right. And in 2003, there was an opportunity of starting a new TB group in Belgium, in J&J. And after Excel Labs, actually, I worked in a company called Eczema, where we were looking at host pathogen interaction. We were trying to target, and that's why I moved to J&J, which was the easiest opportunity for me at that time. Because the group was being set and the molecules were being discovered. And I landed up in a lab of Professor Koen Andries, one of another very, very phenomenal persons I have met. Excel, on one hand, was looking at the broader picture, oncology, kinases, serial entrepreneur. And here was another guy, Koen Andries, who was extremely focused. I learned a lot from him. The biggest thing I learned from him, how to stay focused in this world where we get distracted so easily. And we after 12 years, or maybe 12 to 15 years, we discovered Bedaquiline, and as everybody knows, it changed the human life for millions of TB patients globally.
So I will go deeper into the Bedaquiline story. But before that, a lot of our audience are actually PhD students, and they always have this dilemma whether to, go for an industrial career or go for a career in academia. So how did you make a conscious choice, whether because you joined Eczema, right? Which is kind of a company. So how did you take that decision that I'll not pursue academic research, rather I will switch to industry? What was the decision-making process like?
I think the decision, you never make these conscious decisions. It's the opportunity which shapes you. We all think that our successes are somehow uh, our own, but we don't realize there are so many events and circumstances and people who shape us, who
Absolutely.
shape us what we are today, who give us those opportunities and who believe in us. So it's going from academia to industry. Of course, there has to be opportunity. Of course, there has to be your interest in seeing what you are doing in the labs gets translated. Some people are fascinated by basic biology, which is also fantastic.
Absolutely.
It's how it's how the innovation happens. Some people are more fascinated by the application of basic biology and seeing translation of it. Equally, both are equally fundamentally so important. So I was on the other side. I was trying to see how do I see the application, because I had seen with Excel how Herceptin, the drug he was one of the co-discoverers, how it shaped the lives of the breast cancer patients of the world. You see, you start creating opportunities around the likes of what you want to do in the life,
Absolutely.
and
Absolutely.
they start coming in front of your eyes, and that's why you make those conscious decisions to pick them up and see how you can further establish on those opportunities.
Fantastic. Fantastic. So, I mean, we all know that you played a significant role in the discovery and development of Bedaquiline, which is a groundbreaking, say, drug for the treatment of drug-resistant tuberculosis. So, tell us more about the challenges that you faced during the development of this, and for general audience, also please reiterate on the impact that Bedaquiline currently has on the TB landscape.
Yeah, I think Bedaquiline story is one of the most important thing that could happen to my life. I feel it's sometimes you have to be at the right place at the right time, and to be also guided by your own conviction that you want to make a positive and authentic impact on somebody else's lives. So Bedaquiline is a story like that. We started first drug to be discovered in 45 years for treatment of MDR or multi-drug resistant TB or TB in general. So Bedaquiline got approved in 2012 by US FDA, end of 2012, almost 2013, by US FDA. And what we learned in terms of the innovation was, for the first time, we had a completely new chemical class, which eventually became Bedaquiline. We had a completely new target, which is the ATP synthase, where Bedaquiline targets. We had a completely new clinical development pathway established as we were doing clinical trials with Bedaquiline. And and last but not least, we had developed over last several years an access framework, drug access framework, for distributing this drug in more than 170 countries across the world, and also getting the drug registered in more than 70 countries across the world. So Bedaquiline was transformational in several ways. Now it is part of WHO's treatment regimen for TB, and also on that essential list of medicines list from WHO, both for adult as well as pediatric TB. Manish, I think that it's such a shame. You may you may understand it, because today we have 1 million kids who get infected with TB, and quarter million die. These are young kids. These are young kids, and in spite of all the human progress, we have still 1.6 million people dying of TB every year. So, in spite whatever we call ourselves as a race, there is a huge
But
impact of diseases like TB and diseases like even HIV to some extent, and as well as malaria and several major global public health challenges. So with Bedaquiline, we have been, as I said, that we have been transformational, but it was not me. Let me be honest with you. I am the face of it right now, speaking to you. But there are hundreds of people involved. There are people involved in the discovery teams. There are people involved in the preclinical development, and there are people involved in the clinical development, and there are people involved in access group, who basically what we call lab to the last mile. Last mile is the patient, who bring the drug from the lab to the to the patient.
Mm-hmm.
And the scientific discoveries sometimes are easy, but the most complicated part is to bring the drug to the bedside of the patient. In the And that is where I feel that we have done amazing innovation in not only in the R&D part of it, but also in the access part of it. So right now, with more than 500,000 doses globally have been shipped or used in patients across the world. More than 80% of all MDR TB patients have access to Bedaquiline.
Amazing.
And finally, I think that the key thing which I'm proud of, or how this innovation changed the TB field, is that before the advent of Bedaquiline, all these multi-drug resistant or extreme drug resistant TB patients used to have around 10 to 12 pills every day for almost 18 to 24 months. They used to have a daily injection for almost 6 months. And it was a torture. It's a death threat. It's worse than XDR. Mortality outcomes are worse than any cancer. With Bedaquiline, we have been able to change the treatment paradigm. It's now no longer injectables are needed. It's all oral pills which these patients use. The patients don't have to go to injection of 2 or 3 ml every day to a hospital. They can take it at home. It's all oral therapy. So really, once again, I'm fortunate, I'm lucky, and I'm thankful to so many people who believed in me that we can do it. We can mention that how for the students is the same thing. We as leaders need to believe in people and give them the chance and bring the best out of them. And I think everybody has potential. I believe we all have potential to do.
Fantastic. And I truly appreciate your modesty, Professor Koul. But tell us more about, what inspired you to establish this global public health and infectious disease discovery unit at J&J? And what are the current, objectives and areas of focus for this unit?
Once again, as I said that I'm the face of Bedaquiline, but so many teams. So I read it recently. It's easy to send a rocket to Mars than to discover a drug.
Absolutely. Yeah, it's a whole long process,
It's the whole long process, and I think it's not only the scientists; it's also the leadership. People who have the leadership responsibility in these places, and especially the current as well as the previous leadership of J&J, they really believed in changing the treatment paradigm of TB. They thought that this is possible. We do it with oncology, we do it neuroscience, we do it immunology areas, but TB nobody does it. And and the leadership believed that this is possible, this can be done. And without that commitment, without that support, it's very hard to do any transformational drug discovery, especially for neglected diseases, where the financial incentives is almost nothing. So you need to believe in the human good, in the human health, and the impact your innovation and science can do for millions and millions of people across the world. And that's how the genesis of global public health came into being. Because what we saw as an organization, we saw the impact which Bedaquiline did, and globally we saw it's amazing uh, transformation, not only saving lives of people, that's one thing, but also making it easier for people to take their medication instead of injections and all pill regimen. But the clinic did help to galvanize opinion internally and externally also that you can do transformation for public health problems. You can make a positive change in millions of lives of people. And I think with that commitment, we as a group or as an organization said that we need to increase the scale, go beyond TB. Can we make the similar impact on dengue? Can we make the similar impact on what we call now antibiotic resistance or new antibiotics? Can we make an impact on other flaviviruses or on other bacterial infections? Can we make an impact on COVID with new molecule for COVID therapeutics, not for current, but also for future pandemics? So a lot of things that need to be taken into consideration for public health or public good or the good of human race. And Bedaquiline was the starting point. Bedaquiline was an inspiration, and Bedaquiline showed us as an organization, as well as several other organizations, a way how to galvanize resources, people, inspiration, to tackle some of the very difficult human challenges in terms of the healthcare, as well as in terms of the innovation.
Absolutely. Fantastic. So now I'll come to a very interesting question, because I was a summer trainee at Mtech. And when I saw that, you were appointed as the director of the Institute for Microbial Technology, which is a very prestigious institute in India, I was like all excited. Wow, this is really good, because traditionally what has been happening in India, that it is more of the academic scientist holding this kind of position. And it was kind of a very, I was very excited to see somebody from industry recruited as the director of Mtech. So tell us about that story. How did Mtech happen in your career?
Yeah, it's very interesting. To be honest, when we were launching Bedaquiline, or when Bedaquiline was getting approved globally in 2016, I still remember the day. It was March, somewhere in the March 2016. I had gone to India for the conditional approval or launch of Bedaquiline, and the launch was done by our Honorable Health Minister then, Shri J.P. Naraji, in Delhi. And you remember, I left India in 1996.
Long back, yeah.
And I came back to India, of course, in a different avatar in 2016 on launch of Bedaquiline. And in all those 20 years, I was trying to look for a drug for TB. And this was mesmerizing moment for me. I'm back to Delhi, 20 years. It was a full circle kind of a thing. Coming back started in Delhi as a student, coming back at the Delhi on a launch of a drug where I really worked hard, and also the team and everybody else contributed. So Bedaquiline story was where all my stars aligned together and made that moment of March 2016 possible to see this drug can be used in patients in India, where I came from or where I started with. So after that event, I had a chance to meet our Honorable Prime Minister in Brussels. And that chance meeting also gave me an opportunity to see how we can change the aspects of research we do in India into more translational side. That means how do we galvanize the resources or all the activities in our labs, in the government labs, how that can be used for new products, for new innovations, for new methods to basically improve the public good. So how do we monetize some of the R&D happening in our major public health institutions in India? And that's to cut the long story short. Mtech was another opportunities where all the stars aligned. So everybody aligned together and gave me this fantastic, wonderful lifetime opportunity to lead an amazing institution, as you know. And yeah, it was one of the best phases and one of the best learnings of my life.
And what difference did you see? Like, because you have worked in the, say, the corporate environment for so long, and then, heading an institute that is more driven by academia, although, the aim of it is a council for scientific and industrial research, right? So what did you, what are the challenges that you see ahead of Mtech, and how can one improve upon that?
I think before we go into the challenges, the key is that what are the opportunities?
Exactly. Mm-hmm.
Mtech or CSIR, as you know, is it's one of the very fundamental institutions in terms of if you look at their infrastructure in biopharmaceuticals, whether it's fermentation technology, whether it is GMP facilities, or whether it is deep-rooted biology. I think these institutions are have been built with the aim to deliver what we call industrial research or end products for Indian population in general, or for the global population in general. And Mtech was one of those learnings for me, which where I learned how much possibilities are there for public health R&D to shape the healthcare for its citizens, and it has amazing, talented, aspirational students, amazing infrastructure. One of the most beautiful labs I have seen across the world.
Yeah, it's designed so well. I was also very, yeah.
It's so amazing. It's so amazing. And one of the means very talented people. And when we see in the industry, we realize that there's a parallel set of ecosystem, which is government, and government sees industry, so they don't talk to each other.
Mm-hmm.
We need to be more collaborative with each other. And that's where I saw the possibilities of the government and industry coming together to address some of the global public health issues. We are talking about global public health right now, and it really opened my eyes in terms of how governments can shape and help define the global public health agenda, as well as help us in bringing new innovation for some of these neglected diseases. I think from a challenge perspective, the biggest challenge some of these institutions face is basically the ease of doing science. We build these institutions, but we build the policies which become restrictive over a period of time. Restrictive for students, restrictive for scientists, restrictive in terms of not enabling science. We as leaders need to enable science rather than put up policies which become very difficult and bureaucratic and hard to manage the science. So it's in the end, it's basically a mixture of ease of doing science, ease of doing business in these institutions, facilitating that industry academia interactions, bringing industry closer to the academic institutions, facilitating, helping industry in making that network, and fostering innovation in the end. So I still believe a lot can be done across these institutions. But at the same time, I have now realization our industry also needs to come forward to help and innovate.
Mhm.
Like if you look at countries like Germany, if you look at countries like South Korea, if you look at countries like Israel, very small countries as compared to the size of India or even US for that matter, you see a lot of innovation and R&D funding comes from industry,
Absolutely. Mhm.
like to the tune of 80 to 85% in some cases. While in India, it is the reverse. A lot of expectation is that the government spends on the R&D and the science budget, while industry is basically look at not that participatory in the early innovation part of the whole. So we need enablements where we have more industry participation in the R&D and I'm sure that the day we have that in India, it will change a lot of R&D ecosystem as well as create more opportunities for science to foster and also kill some of those restrictive policies and enable students to do better science.
Fantastic. So now you're also an adjunct professor of translational research at the London School of Hygiene and Tropical Medicine. So how do you bridge the gap between academia and industry in your work? And what are some of the strategies that you employ, say, to foster innovation and collaboration in the field of drug discovery? Say,
Yeah, I think London School is the outcome of Mtech learnings. So Mtech learnings is told me basically that what public health institutions, how they can help and shape the global public health agenda and help also in innovation part, especially the early drug discovery innovation part of it. And so London School is the third most reputed public health. Its ranking is around second or third globally in the public health domain. One of the amazing, talented institutions in the world. So what we did is that we set up a TB lab, which I run right now with 5 to 10 people, and this is on top of my J&J job. So I have those two hats right now.
and that's why the question: how do you bridge the gap? But yeah, go ahead.
Yeah. So you have to be at different places at different times. Somehow I'm managing it, but I think the mission is same, irrespective of the domain. The mission is same. How do you bring the next generation TB drugs? How do you change the TB treatment paradigm after Bedaquiline? So that's how London School is happening. But beyond London School, we also have established labs in Duke and US, in Singapore, where we are looking at flaviviruses like dengue, Zika, Japanese encephalitis, yellow fever, and few other things. Very nasty viral pathogens. And then we have another lab in a very talented environment of X3D, which is in University of Cape Town, looking at antibiotics and drug resistance from an African perspective.
Mm-hmm.
So trying to discover new drugs and new molecules, new antibiotics, taking into account the emerging resistance to antibiotics in an African continent. So very, very nice innovative aspects of a public-private partnership. All the outcomes of learnings from Mtech, learnings from Bedaquiline, learnings from lab in Max Planck, and of course, learnings from Delhi University.
Amazing. Yeah, coming back to, to focus on our listeners who are generally like, aspiring, say, students, PhD students, master students, and they all write me, I mean, especially when they kind of see podcasts like this, they ask about, how could they develop like the key skills and qualities that are perhaps, say, required to be a successful scientist in, say, in your case, in big pharma. And what advice would you give to the students, in terms of skills? How can they upgrade their skills so that one day they can be a successful scientist in, say, a pharmaceutical industry?
I think the key is to understand that biology is fundamentally changed.
Mm-hmm.
Biology is more interdisciplinary. Biology is more not the same biology which we used to do, you and me used to do 20 or 30 years ago. It has more the components of informatics. It has more components of the technology. It has components of physics, chemistry, all coming together. We are right now in an amalgam of biological sciences with all different domains of sciences coming together.
Absolutely.
This century is going to be the century of biology. I have really firm belief in that. Whether it's the application of data sciences to the human health,
Mm-hmm.
whether it's the application of all the genomics to the human health, we are going to unravel amazing aspects of our own being, our own consciousness, I hope one day, and also solve some of the major mental health issues what we have, right? From Alzheimer's, Parkinson's, and get deeper inside our brain in our own understanding of what we as human race are.
Mm-hmm.
So for students, I think the key is that you are authentic to what you are trying to do or what you believe in. That authenticity is very important. And also embrace the opportunities, as I said, that in the multidimensional biology area.
Mm-hmm.
From pharmaceutical industry, we employ people right in the regulatory sciences. It's not only biological wet lab work which is critical. Regulatory sciences, pharmacology, toxicology, ethical sciences.
Market
Even,
access, as you said.
market access is one of those things. And discovery biology is one of those things. Even clinical sciences. So there are so much opportunities which a lot of us are not really aware of, and we go with a one-dimensional thinking that this is the start and this is the end. While there is no end to the learning and there is no end to the opportunities also. You just need to keep open mind. You just need to be looking. You should not, somebody, as Steve Jobs said, or somebody else, not he didn't say it, but somebody else said that stay hungry, stay foolish.
Yeah, that's Steve Jobs. Yeah.
Yeah.
Stay hungry, stay foolish. Yes.
Yeah. So I think that's how you grow. You in the moment you start thinking that this is the line and this is the field. It means you don't evolve. You have to constantly evolve with the changing pace of technology, changing pace of science, and there are multiple of opportunities. There are so many opportunities for all these young, talented biologists which are out there. I feel that the field is so open. It's the best time to be in the biological sciences.
That's inspirational. Yeah, that's amazing to hear.
Because when you see how siRNA is, not only siRNA, CRISPR.
Absolutely. I was about to say that, like when you were talking about jumping genes, because in my days, siRNA was a big thing, and then now when I talk to new PhDs, they all talk about CRISPR and, gene editing and everything. And, it just excites me, how biology is getting developed day by day. So that's amazing. Yeah.
Yeah, we are using the CRISPR-I right now, even in our labs, to knock off genes in the microbacteria. It's such a powerful, amazing tool, and the application of the CRISPR-I in sickle cell anemia and so many other cell and gene therapy applications. It's really, really amazing. I think the way the oncology is changing right now with cell therapies, with
Gene and cell therapy is absolutely CAR-T
is huge transformation. We are living at the cusp of technological and biological immune oncology. Wow, this is somebody we never thought this is going to happen, but now we are treating cancer by boosting our own immune systems. Think about it, where we have come from and where we are going in terms of eradicating some of these diseases. And I'm sure that one day we can get inside our brains a little bit better, understand Alzheimer's, Parkinson's, and even our own, how does our consciousness arise and mean? And it will be in the end a biological phenomenon, hopefully.
Yeah, I mean, Elon Musk has a startup called Neuralink, and he wants to build something, I mean, it's not a startup anymore. It's already a legit company, working on, say, connecting the brain with external neural links. Uh, I mean, you have partly already answered, but I was going to ask you about how do you see the future of drug discovery, and are there any emerging trends and technologies? You have touched little on them, but perhaps you can elaborate on them.
Yeah, I think in the last 3 to 4 weeks, uh, we looked at 3 or 4 companies who are doing AI based drug discovery.
Mm-hmm.
If you would have spoken to me 6 months ago, I would have said, this AI based drug discovery, this may not happen because drug discovery is so complex and so challenging. It needs experience, it needs expertise, it needs different things coming together, and it's not in silico.
Mm-hmm.
It's more a wet lab biology and driven or chemistry driven focus. But now I will take that I am wrong. I feel that that our knowledge is get outdated very soon. So this AI role application in drug discovery is basically looking at algorithms which can give you new chemistries based on just the crystal structure of any protein. And that novelty, without going through the high throughput screening route, seems to be very exciting for me, especially for global public health, where the innovation is lacking.
Mm-hmm.
Innovation has stayed same and has not moved at the same pace as we have seen in the oncology or immunology or other therapeutic areas. So once again, amazing in terms of what the possibilities are and how cost effective they can be. It still needs to be proven. Let me be honest. Maybe my enthusiasm is misplaced, and a lot of these things still need to be proven. But we are already using data science,
Big way, I should say.
big way in
Yeah.
big way globally. Data science integration in clinical trials, in patient recruitments, in finding those clinical sites, in defining those biomarkers, and data science in the early discovery biology, whether it's the omics platform or it is the integration of genomics on a high dimensional patient level, is or even the high content imaging story is really, really transformation that you can image inside the cellular processes.
Wow.
Never thought about. Now,
Said now biology is not just biology alone anymore. It's because you have so much of advancements in other sciences as well. You can tackle the most difficult questions in biology with the tools that you already have. So that's
yeah,
amazing. Yeah.
I think the only what we have to be cautious about is that we don't become mere applicators of the tools.
Absolutely.
We really need to keep our fundamental understanding and power of data analysis is the most critical thing here. It just doesn't need to be that we use these fancy tools for the sake because they are there. The questioning, the answers,
Problem solving. Yeah.
the problem solving is the most critical thing. Sometimes this most complex things get solved by very simple means with very simple understanding. So we should not also get swayed away by the technology prowess. We should keep our common sense and common inquisitiveness still alive. That's what science is in the end. It's basically, we may not have the full knowledge, but if you give me a data set, I may be able to understand and interpret the most critical aspect of that particular data and see what the application lies.
Fantastic. So coming to the final section of the podcast, what advice would you give to biology students? If you were to give one great advice to biology students who want to have a career in this path, biotechnology, what would you give?
I think my main advice is basically stay focused,
Mm-hmm.
stay authentic, authenticity is key in your relationships, in your approach, in your career choices. Stay believe in what you are trying to do. If you don't find the purpose, you don't need to do it. There are everybody is smart enough to find a way around this world. So that and don't take shortcuts. I think sometimes we think that the short-term gain will give us a long-term success. It's not true. We need to develop that deep expertise and deep talent, and success is always there along the way. If you have authenticity, if you are absolutely.
So, Professor Koul, tell us also about your hobbies. What do you do when you do not do science?
Yeah, what I do when I don't do science is basically I love badminton. These days, I've been doing it for the last 20 years, but these days more specifically because it helps me. Those 2 hours help me to focus on the shuttlecock.
Wow.
It declutters my mind quite a lot. So it takes the toxic end products of my neural metabolism out, it's a phenomenal thing that helps me manage the stress of everyday life.
So badminton, yeah. By the way, people who play racket sports tend to live longer. So that's a side effect that you have.
Yeah, I don't want to live long, but
yeah, I think the society will be happy to have you for long, because you keep on contributing with your fascinating innovations. Apart from badminton, what else?
I think just to the previous question, the key is also the social interactions. I mean, there's a lot of data around it. The more positive interactions you have, the more interactions based on that social network, which is based on enablement, and while you surround yourself by people who are inspiring, who motivate you, who bring you up, and with whom you can share so many things, and that is the reason of long life and longevity.
One of the that's
one of the reasons.
Yeah, but that's one of the most vital indicators. It's actually there's a very long study of 75 years, longitudinal study done in Harvard, and that exactly says what you just said.
Yeah, so. and beyond that, I think I also like cooking. I love Indian food, and several other international cuisines also, but particularly in the North Indian and South Indian. South Indian is my favorite.
Wow. And mine too, by the way. Mine too. I love dosa and idli, and especially I go to Frankfurt just to have that because I don't get in my town.
I tried to add dosa the first time at home. It was disaster.
Yeah, it's so tricky. It's so tricky. Dosa is tricky.
It's tricky. Yeah, beyond that, I love to read, but in the last several months, nothing much. But I really like reading a lot. So it's also one of my favorite passions to read. Yeah,
fantastic. So, Professor Koul, thank you so much for sharing your insights with us today. It's been a pleasure talking to you.
Thank you, Manish. It was wonderful talking to you. Hope to see you sometimes in real life.
Yes, and you should visit me sometimes. Whenever you are this side, I live in Mannheim, so you're most invited to join me at my place.
Same here. If you are close by, happy to host you.
Thank you so much. And to the audience, thank you so much for tuning into Biology Career Insights. Don't forget to subscribe to our podcast to get the latest episode delivered straight to your device. See you next time. Bye-bye. Thank you.
Bye-bye.